Hello students, in a continuation of the last video, we'll start with part 2, unit 2 of the paper Biomolecules and Microbiology, and this video deals with structural organization of proteins. As we all know that the structural organization, we have 4 levels of protein organization. In that first one, it is primary structure. Protein primary structure is the linear sequence of amino acids in a protein. It is a linear sequence of amino acids. So by convention, the primary structure of a protein is reported starting from amino terminal. It is also called N terminal, and end with carboxy terminal. It is also called C terminal. Fixed number of amino acids are arranged in a particular sequence. So if you know the complete sequence of a polypeptide chain, what is the amino acid present in N terminal and which is the amino acid present in C terminal. So all these will depict the primary structure of a protein. A fixed number of amino acids are arranged in a particular sequence. So this is called primary structure. The sequence of amino acids in protein determines its biological role. Different proteins have different sequences. Therefore, the study of total number of sequences of amino acids in a protein is the primary structure. So this picture I have already shown you in the previous video also. So this is the structure of insulin, where you have A chain and B chain, and the number of amino acids in A chain and position of disulfide linkages, it depicts the primary structure of a protein. Then what is the next level of protein organization? It is called secondary structure. Secondary structure is the conformation of protein, means it is a shape. Conformation of a polypeptide chain by twisting or folding is referred as secondary structure. There are two types of secondary structure, that is alpha helix and beta pleated sheet, and you have one more structure that is called bends. We call them as beta bends. In secondary structure, the first one, it is alpha helix. If the polypeptide chain, it takes the helical form, it is called alpha helix. So this structure was proposed by Pauling and Carey in 1951. The polypeptide is twisted or coiled to form a right-handed helix. So polypeptide chain is twisted to form a helical structure. The distance between each turn, is 5.4 angstrom, and there are 3.6 residue. There are 3.6 residues per turn, and the spacing was 1.5 angstrom. The R groups are seen protruding out of the helix. R groups are nothing but the side chain of different amino acids. As I shown you in the previous video, there are 20 different amino acids. They have the different R groups. Okay, these R groups, they protrude outside the helical structure because they don't have the space to be too fixed inside this helical structure. So this structure is stabilized by an intra-chain of hydrogen bonding, wherein hydrogen of NH group combines with oxygen of CO group, that is NH and CO. So there is a role of hydrogen bonding which stabilizes this helical structure. So every peptide group participates in hydrogen bonding. So this type of structure is found in many proteins in combination with the other structure, and it forms with a low free energy inside the cell. The next secondary structure, it is called beta pleated sheet, where the polypeptide chains are arranged in the form of sheets. The polypeptide chain is not helical, but it is a zigzag sheet-like arrangement. The distance between each turn are between the amino acid, it is 7 angstrom, and the polypeptide chain are arranged by side in the form of pleats. Okay, so here if they run from N to N terminal or N to N terminal, they are in the like groups, they are called as parallel beta pleated sheet. If they are with opposite groups, N, C, they are called, that is N terminal, C terminal, N terminal and C terminal, opposite direction, they are called as antiparallel. So here the polypeptide chains are arranged side by side in the form of pleats. There is inter-chain hydrogen bonding between the chains, and each peptide bond participate in the hydrogen bonding. The chains are arranged parallel or antiparallel to each other. So example for this, it is the structure of silk fibron is the beta pleated like structure, where it is very that is a tough structure of protein because of its pleated like arrangement. The silk emitted by silkworm consisting of two main proteins, serine and fibrin. The fibrin being the structural center of the silk. The serine is being the sticky material surrounding it. Fibrin is made up of repetitive structure of amino acids, that is glycine, serine, glycine, alanine, glycine and alanine. And this forms the beta pleated sheets. That's why it is insoluble and it is used for the formation of silk material. It is an insoluble protein comprising of filaments of raw silk fiber. That is why it is very tough and insoluble because of its structural arrangement. The amino acid composition of silk fibrin from the Bombyx mori consisting of mainly glycine 40%, alanine 30% and serine 12%. The right-handed alpha helix and beta pleated sheet are common structural motifs found in most of the proteins. They are held together by hydrogen bonding. That means these structures are stabilized by the hydrogen bonds along with the other weak force of interaction that I'll tell you, and the amino acid, that hydrogen bonding between the amino group and carboxyl oxygen within the amino acid backbone, which run in the form of a chain. The other secondary structure include turns, loops, hairpins and flexible linkers, because when the alpha helical structure runs like this, they have to interconnect with the next that alpha helical structure. These regions we call them as turns. So there are many different classification of turns within the protein structure, including alpha turns and beta turns. Typically contain 4 amino acid residue, proline and glycine. Because of their small size, they are commonly found in the turn motifs, as the cis configuration of proline favors the sharper conformational bends, while the minimal glycine side chain, that is R group of glycine, is very small, and the chain allows for tighter packing of amino acid to favor the turn structure. So these are the less organized structure. So these linking are when these beta pleated sheet or alpha helixes, they take turns. So these regions we call them as turns, loops and hairpin structures. They are also considered as secondary structure of proteins. The next level of organization, it is tertiary structure. The overall three-dimensional structure of polypeptide is called as a tertiary structure. The tertiary structure is primarily due to the interaction between the R groups of amino acid. The secondary structure, it is only the hydrogen bonds which plays a major role, but in the tertiary structure, R groups come into play that make up the protein, that R group interaction that contribute to tertiary structure. So special type of covalent bond that can contribute to tertiary structure is called a disulfide bond. So this is the schematically tertiary structure of a protein. It is shown. The helical form of polypeptide folds into spherical, globular, ellipsoidal or other conformation. Conformation means shape, which is called tertiary structure. This folding is necessary for the biological activity of proteins. The example for this, the enzymes, this is a structure of myoglobin, immunoglobulin. So this here you can see in the myoglobin there are 8 helices which forms a tertiary structure of a protein. And here also I have shown you the hydrogen bond, ionic bond and disulfide linkages, van der Waals force of interaction, which contribute to the tertiary structure of a tertiary structure. It includes hydrogen bonding, ionic bonding, dipole interactions and dispersion forces, basically non-covalent bonds. The R groups, which like charges, they repel one another, while those with opposite charges can form an ionic bond. Similarly, polar R groups can form hydrogen bonds and the other dipole-dipole interaction. All these forces which contribute to the 3D structure, that is tertiary structure. The last level of protein organization, it is a quaternary structure. So many proteins are made up of a single polypeptide chain, but have only 3 levels of structure. Only if it is made up of only a single polypeptide chain, it forms a tertiary structure. But some proteins are made up of subunits, multiple polypeptide chain known as subunits. And when these subunits come into together, they gives the protein its quaternary structure. Example for this, it is a quaternary structure. It is a hemoglobin, where you can see here 2 alpha chain and 2 beta chain, where the heme group, it is in the center of this that is globin structure, and where here this hemoglobin, it is situated in the center of these red blood cells to carry the function of oxygen. Hemoglobin is one of the most widely studied proteins. Its structure consisting of 4 subunits, 2 alpha and 2 beta tetramer, and each containing a heme moiety with iron in the ferrous state. Each alpha chain consists of 141 amino acid residue and beta chain of 146 residue. Each hemoglobin is in blood carries the oxygen from lungs to the lungs or gills to the rest of the body. So there it releases the oxygen to permit aerobic respiration to provide energy to power the function of organism in the process called metabolism. So here this slide explains you there are several different types of forces which contribute to tertiary and quaternary structure of protein. The first one is covalent bonds. Covalent bonds, the peptide bonds between the amino acid, that is CONH bond, it is also contribute its force, its part in the formation of tertiary structure. Disulfide bridges which between the cysteine residue and non-covalent forces, that is hydrogen bonds, that bonds are present throughout the protein. The bonds in the middle of the protein structure contribute most to the stability, as they are more away from the water, and they keep the molecule away from the water. And van der Waals forces of interaction, a short range dipole-dipole interaction between the close atoms, and pi overlap, pi electron cloud delocalized over the rings and bonds, and electrostatic bonds, ionic interaction, salt bridges between the residue. All these forces, they contribute to the formation of tertiary and quaternary structure. All these forces can be broken by the change in the pH and high ionic strength. So this I have shown you the flow chart of primary structure, where if you enlarge a polypeptide chain, you will be knowing the sequence of amino acid starting from N terminal and C terminal. You will be knowing which is the first residue, which is the second residue, all this. The primary structure, sequence of chain of amino acid, it is nothing but a primary structure. Next one, the second conformation, where they can fold in the form of sheets or helix. So that is a secondary structure, beta pleated sheet and alpha helix. In the combination of these two are only the alpha helical, they can form a tertiary structure, where you will get a biological function, the three-dimensional structure of a protein. It is called tertiary structure. And the last one, association of one or more subunits, you will get a quaternary structure. So primary, secondary, tertiary and quaternary structure. This is how the proteins are organized. And there are two main classes of protein tertiary structure, that is fibrous and globular. Fibrous proteins are generally composed of long, narrow strands while structural roles. So these fibers and sheets are mechanically strong or water insoluble. Globular proteins generally have more compact, rounded shape and have a functional roles. So this is the table, shows the difference between globular and fibrous protein. Globular proteins, they are compact protein structure. Fibrous proteins are extended protein structure. Globular proteins soluble in water and in lipid, insoluble in, these proteins are insoluble in water. Third one, secondary structure is complex with mixture of alpha helix, beta sheet and loop structure. Secondary structure is simple based on one type, and quaternary structure is by non-covalent forces. Quaternary structure is usually together held by covalent link bridges. And functions in all aspects of metabolism, they are present in the form of enzymes, transport proteins, immunoglobulins with the immune response and immunoprotection and hormones, and functions in structure of the body, silk fibrin, ligaments, hair, silk and collagen. So this is the structure of myoglobin, and this is the structure of collagen, which comes under fibrous protein structure. Okay, so in this video I told you about different levels of organization. We'll continue in the next video. Thank you for watching.